Today, our topic of discussion is unraveling the significance of DNA methylation in epigenetics. First of all, let us discuss what is epigenetics and its explanation. For example, epigenetics is the study of changes in gene expression that do not involve in the alteration of DNA sequence itself. For example, we can say that simply we can say that if a DNA sequence get altered, so the gene sequence or the genome or the expression get altered. Without alteration in DNA sequence, how it is possible that our gene, our genome get altered? That's very interesting question, for example, for scientists, and the whole process of epigenetics is discovered for this fact. The jargon of epigenetics, the processes of epigenetics involve the DNA methylation, histone modification, and non-coding RNA molecules. For example, by the help of DNA methylation, histone modification, and non-coding RNA molecules, we can explain the epigenetics, how the genome or the gene sequence or the gene expression or whole genome get altered without alter the DNA sequences itself. Okay, let us relate a diagram of the epigenetics. For example, in this process, we can say that a human cell consists of bone cell, liver cell, or the muscle cell. The part of, for example, skin cell involves some promoters and the whole DNA sequence, whole DNA region. Liver cell have the promoter region, muscle cell have the promoter region. Okay, now the question is that the DNA sequence is same for all cells, then why skin cell express different genome than that of liver cell, and liver cells express different genome than that of muscle cell? For example, muscle cell involve different in function than that of liver cell perform different function than that of skin cell. The protein production in skin cell is different than that of liver cell, and the process, protein translation or transcription, is different in that of liver cells than that of muscle cell. How this whole process can take place after the fact that the DNA sequence of both three cells are same? That the processes of epigenetics, that we add some modification, some interesting facts which include maybe DNA methylation, maybe histone modification, it may be RNA non-coding region modification. It involves this process that only a specific sequence, only a specific region which is called exome region of the muscle cell get sequencing and have the specific protein for its own function. For example, liver cell have its own cell or its own function, its own coding region, and which is maybe defined by some modification, either the DNA methylation, by either by histone modification, or maybe RNA non-coding modifications. Next, let us relate the DNA methylation, which is my exact topic of discussion. What is DNA methylation? Exactly, हम क्या करते हैं? We are included the methyl group, for example, CH3 group at the region of cytosine. We add the methyl group at cytosine. We take a methyl group from SAM and we add that group in the carbon of cytosine, or make the 5 methyl cytosine by the process of methylation, DNA methylation process. For example, we can relate this DNA methylation by three enzymes: DNA methyl transferases. It may be the DNA methyl transferase 1, DNA methylase transferase 3A, DNA methylase transferase 3B, which are abbreviated as DNMT1, DNMT3A, and DNMT3B. We have some CPG Iceland, which contains the CPG sites. At these CPG sites, we add the methyl group, which is called DNA methylation. We uses the DNMT3A, DNMT3B to add that methyl group at the region of CPG site. By the addition of these CPG site, methyl group at CPG site, we also retain that methyl group at the CPG site. So we use the DNMT31 as the arrow indicated. Next, by the addition of CPG sites at the embryonic cell, we can say that when the skin cell differentiate, it all the DNA sequence have that methyl group and that retained by DNMT1. And now, how this DNMT3A and DNMT3B at the methyl group at the CpG site, which are the part of CpG Iceland. This methyl group is taken from SAM, and that methyl group is added to the carbon number 2 of the cytosine, which make the 5-methyl cytosine, and that's the whole process by which DNMT3A and DNMT3B added a methyl group from the SAM at the cytosine group and the cytosine DNA nucleotide at the CpG site. Now, how this whole process is taken, let's take an overview. For example, first of all, we change the propagation of cytosine and it is inverted at 180 degree. DNMT flip the cytosine at 180 degree and SAM and get a methyl group from SAM. DNMT obtain methyl group from protein SAM and methylated the cytosine is flipped back. For example, that whole process is taken by DNMT3A and DNMT3B and it is retained from embryonic cell to the differentiated cells by the help of DNMT1. Understanding the role of DNA in epigenetic modification. For example, if there is a skin cell and only a specific sequence, only a specific region of that skin cell DNA must be translated or translation, so then we add a methyl group at that region. So only that region get translated or transcription and translation of only that sequence, that specific region can be done, and protein of our desired process, our desired function can be formed. That's the role of DNA methylation in epigenetic modification. For example, DNA methylation is just like a methyl at the methyl group, and that methyl group is just like a marker that we have that DNA sequence in our translation process or not, or we want that sequence in our translation process or not. That whole process is done by DNA methylation, or that only a specific region can be modified to get translated so the epigenetic process can be understandable. Implication of DNA methylation in gene expression. Aberrant DNA methylation pattern have been linked to diseases. For example, if DNA methylation or a methyl group, which is we use as a marker, is added to a wrong region, so it cause the translation of that wrong region, and if the translation get occurred of that wrong region, so the disease can be happened, and like cancer and other diseases in which there is a uncontrollable deviation or uncontrollable mass of unwanted protein, and that protein is encoded by a aberrant methyl group which is added to a aberrant DNA sequence. Epigenetic inheritance. Current research and discoveries in DNA methylation, in advancement in epigenetic sequence technology, we are unable of mapping the DNA methylation pattern, fueling further exploitations and expanding our understanding of this complex epigenetic mechanism. How a gene get translated or transcription without any apparent DNA? All that process is done by the DNA methylation or DNA modification in the epigenetic process. For example, like this process, we have a cancer cell in which a hypermethylation or hypomethylation is done. For example, methyl group are added at a wrong region which can't be translated, which we want can't be translated or can't be transcribed to form a specific type of protein. So if that protein, that sequence get translated or transcribed, so we can say that an aberrant protein can be formed, and that aberrant protein mass causes the cancer cell. Thank you.